Good morning to the examiner and everyone present. My name is NUREEN ALIA BINTI NAZARUDIN, and today I will be presenting my research proposal titled Association between Mitochondrial DNA Polymorphism and the Risk of Metabolic Disorder for Diabetes. My supervisor for this research is Dr. SERI MIRIANTI BINTI ISHAR. To introduce this topic, type 2 diabetes mellitus (T2DM) is a massive global crisis. Currently, 589 million adults have T2DM, and this is projected to reach 853 million by 2050. Locally, Malaysia has one of the highest prevalence rates in Asia. Good morning. Good morning to the examiner and everyone present. My name is NUREEN ALIA BINTI NAZARUDIN, and today I will be presenting my research proposal titled Association between Mitochondrial DNA Polymorphism and Risk of Metabolic Disorder for Diabetes. My supervisor for this research is Dr. SERI MIRIANTI BINTI ISHAR. To introduce this topic, type 2 diabetes mellitus (T2DM) is a massive global crisis. Currently, 589 million adults have T2DM, and this is projected to reach 853 million by 2050. Locally, Malaysia has one of the highest prevalent rates in Asia. While we often focus on lifestyle factors, we must also look closely at our cells. Mitochondria act as a cellular powerhouse, producing the ATP required for our pancreas to secrete insulin. If the mitochondrial DNA or mtDNA is mutated or altered in its copy number, it can severely disrupt this energy production and lead to metabolic disorder. This brings us to the problem statement. First, lifestyle and obesity do not fully explain why some individuals develop T2DM while others do not. Second, establishing a definitive genetic map is difficult due to heteroplasmy, which is the mixture of both mutant and normal mtDNA existing in a single cell. Finally, there is a massive ethnic data gap. mtDNA variation is strictly maternal and highly ancestry dependent. Yet, localized genetic profiling for Malaysia's multi-ethnic populations remains heavily under research. To address these, I have three main objectives. First, to identify specific mtDNA variants associated with T2DM risk. Second, to evaluate the biochemical mechanism triggering this metabolic dysfunction. And third, to review population-specific differences to identify evidence gaps relevant to Malaysia. Correspondingly, my hypotheses are specific variants like A3243G and D16189C significantly increase the risk. These polymorphisms impair ATP production and increase oxidative stress. And finally, these associations will exhibit distinct genetic risk profile across Malay, Chinese, and Indian populations. The significance of this study is to provide a consolidated reference and comprehensive guide on high-risk genetic targets for Malaysian clinical researchers. Ultimately, this lays the foundational template to develop localized, ancestry-specific precision medicine, replacing the failing one-size-fits-all screening with targeted prevention strategies unique to Malaysia's diverse demography. Diving into the literature review, we found that pathogenic variants disrupt oxidative phosphorylation. When this happens, ATP production abruptly halts, leading to a severe leakage of reactive oxygen species, ROS. This induces cellular AR stresses and causes irreversible beta cell apoptosis. Specifically, the M3243A2G variant acts as a massive universal risk factor with an odds ratio of 3.45, while variants like D16189C are highly ancestry dependent. Furthermore, a decreased mtDNA copy number in the blood acts as a strong independent biomarker for incident T2DM. Interestingly, from a forensic application standpoint, high mtDNA copy number helps in degraded DNA cases. Furthermore, a reduced mtDNA copy number can act as a biomarker to indicate a history of T2DM postmortem, which is highly useful when medical records are unavailable. For my methodology, I conducted an extensive database search using platforms like Google Scholar, ScienceDirect, and PubMed. Literature reviews are then managed by Zotero, and then synthesized data are systematically tabulated into a standardized 5-column format to easily compare the findings. So these are the references that I used for this slide. Thank you for your time, and I welcome any questions from the panel.